Tessier is from the University of Michigan in Ann Arbor, Michigan. His project is called the Efficient Brain Delivery of Neuroprotective Antibodies in Alzheimer's Disease using a number I won't say, biospecific shuttles. And so he is also receiving a $300,000 award this year in honor of Dr. John Krojanowski. We're naming it the John Krojanowski Memorial Award. Let me tell you a little bit more about Dr. Tessier. He is currently the Albert Maddox Endowed Professor in the Department of Chemical Engineering, Pharmaceutical Science, and Biomedical Engineering at the University of Michigan. He got his PhD in chemical engineering from the University of Delaware, was a NASA graduate fellow. He did his postdoctoral studies at Whitehead Institute for Biomedical Research at the Massachusetts Institute of Technology as an American Cancer Center fellow. And he served as a full professor at the Rensselaer Polytechnic Institute prior to moving to the University of Michigan in 2007. His research focuses on designing, optimizing, characterizing, and formulating a class of large therapeutic proteins, antibodies, that hold great potential for detecting and treating human disorders ranging from cancer all the way to neurodegenerative diseases. He's received a number of awards: the Pew Scholar Award in Biomedical Science, the Humboldt Fellowship for Experienced Researchers, the Young Scientist Award from the World Economic Forum, the Young Investigator Award from the American Chemical Society, and finally the Career Award from the National Science Foundation. We're very blessed to have Dr. Tessier as one of our grant recipients this year. Please come up and tell us about your project.
All right, well, thanks so much. It's really such a pleasure and an honor to be here today. I've really enjoyed last night and today getting to know you, and you've made a very strong impression on me. I noticed a few things. First of all, you are very gracious people, just very warm and welcoming, and I really appreciate that. And you're also very generous, of course. And I noticed you're also very passionate about Alzheimer's disease and advancing research, and that comes through very clearly in my interactions with you. And frankly, it's inspiring. And so it really means a lot to be here, inspired by you, and really honored to be part of this very special event, and really humbled to receive this award. So as was mentioned, I'm coming from Michigan, where we don't have warm weather yet. So this has been a very nice surprise here, I can tell you that. So enjoying that very much. I want to take a minute here and introduce my team. It's a team of 4 people, and in no particular order, I'm going to go first because my part of what we do is I make molecules. So the molecules that I'm going to tell you about today, we make those in our lab and we design them and produce them for getting into the brain and doing things that are neuroprotective in the brain. Our second team member is Colin, and Colin is an expert in what we call pharmacology, and he's an expert at tracking where molecules go in the body, the concentrations in the body, and ultimately in the brain. And that's really important because if things are going to be effective in the brain for treating this disease, we need to know the concentrations, where they are, how they distribute. The third member of our team is Jeff, and Jeff is an expert in animal models of Alzheimer's disease and understanding whether molecules we get into the brain impact their ability to think and remember and do tasks as a way to evaluate whether these molecules potentially could be effective in humans. And then finally, the last member of our team, last but certainly not least, is Henry Paulson. You might remember we used to have a Treasury Secretary, Henry Paulson. This is a different Henry Paulson. He goes by Hank, and Hank is an expert in many things related to neurodegenerative disease, including he figures out the pathology of the brain and whether the molecules we're getting into the brain are improving brain health and protection. He's also the director of a very large Alzheimer's disease center at the University of Michigan. So he is really helpful for us to provide a big picture view on the potential impact that this research has on actually treating this disease. So I want to have a little bit of fun, and I'll try to make this pretty informal. Think about, we're trying to solve two problems. Frankly, if we made progress on either problem, it'd be a pretty big deal. And if we could make progress on both problems, that'd be even a bigger deal. And so the two problems that we're trying to tackle, one is the blood-brain barrier. We heard a little about that. How do we get molecules into the brain in a predictable and efficient manner? So we'd like to be able to do it so that every time, every new drug we have, we could get it into the brain in a predictable manner at concentrations that we could predict beforehand, so that whether the molecule we deliver or not turns out to be the final drug, we would have a mechanism where we could deliver any drug we're interested in to the brain in an efficient and predictable manner. So it's helpful when we think about this problem just to think for a minute about the brain. And I don't have to convince you, the brain sure is a fascinating organ. And one thing about it is that it has an incredible amount of blood vessels. So it's been estimated there's like 400 miles of blood vessels in the brain. So this is what we say is highly vascularized, or there's just blood vessels everywhere. And if we zoom in and thought about what one of these blood vessels look like, so here is a blood vessel that we can see there's a big cell in the middle of the blood vessel. It's a cross section. In this tan region, if you could see it sort of just on the, just the inside of this tube, that's the blood-brain barrier. And it's actually just a layer of cells. So actually the blood-brain barrier, really, the key part of it is only one cell thick. And the question is, how do we get through that cell, particularly for large molecules? So we are particularly interested in just natural immune proteins. So antibodies that are normally circulating our blood. During COVID, we've heard enough about antibodies, right? We've heard about, and so in the blood, we have thousands of different antibodies that do different things. And we're interested in delivering antibodies to the brain that would be neuroprotective. And so what we need to think about is, how do we get antibodies that if we inject them into the bloodstream, they circulate and they go to the brain, how do we get those molecules to move from the blood into the actual brain? We call this the brain parenchyma, or the region outside that would actually be able to do something useful, like target a neuron to protect it against disease. So this problem actually could be reduced to this very simple picture, and there's just two of what look like those are membranes. That's just one cell layer thick. And this is really an aspirational picture of what we'd like to happen. So we'd like to add antibodies. These are these sort of Y-looking structures. We'd like to be able to add those, just give a shot, have them circulate through the blood into the brain. Then we'd like them to move across into the brain. But the problem is that most molecules don't do that. And once in a while you get a molecule that does, but the problem is it's very unpredictable. And so for example, the Alzheimer's drug that is approved, the antibody that is approved, it does seem to get into the brain some. That's how it has an activity. The problem is it's not predictable which ones are going to do that and which ones are not, which really impedes the development of new antibody therapeutics. Okay, so I want to show you one picture, and now this picture is intentionally busy, but don't worry about the details of why it's busy. But imagine this is the blood-brain barrier again. This is just another picture where each square here is a cell, and this is the blood side and this is the brain side. And what I want you to take away from this picture is the brain needs lots of nutrients. There's lots of things going, there's a lot going on up there. I mean, maybe some of us more than others. I would be a less than others kind of person, but the point is, there's an incredible amount of energy that we need, and therefore there's a lot of nutrients we need to pass into the brain. And so that's what you can see here, is that even though you can't see the details, you can see these little colored things. And those are just different kinds of nutrients. So for example, one area that people have focused a lot on is the fact that we need things like iron in the brain. Okay, it's an important molecule that the brain cells need. They need iron. So one approach to try to get molecules into the brain is to kind of piggyback on iron transport. Now, the details of how that's done is beyond the scope of today, but there's proteins that carry the iron into the brain. So if you could get your drug to want to attach on to one of these proteins that's carrying iron into the brain, that actually could be an attractive way to make a molecule so that you know it's going to get into the brain, because it'll bind to that iron transport protein. The iron transport protein does what it does. It crosses the blood-brain barrier and then releases iron on the other side. Now, this approach has taken a long time, but it's actually now in clinical trials, and there's a lot of exciting things going on in that space. So this really looks like it has potential. Now, we are going to focus on, in this project, a different approach. So the approach that is under the arrow is we're not going to try to piggyback on iron transport. We're going to try to piggyback on amino acid transport. So amino acids are just the building blocks of proteins, and there's a lot of protein synthesis going on in the brain. So there's a lot of need for the building blocks to get transported into the brain to mediate the synthesis of proteins. Now, there's some interesting reasons why we think this mechanism is really attractive. And I'm going to explain and just show you some data that it looks like it may be more effective than the bit of data. I'm not going to try to freak you out, but there is a couple pieces of information that are really useful to say, does this make sense? And animal models are important for a lot of different reasons. But one of them is that when you inject something into the bloodstream, there is a lot of things that can happen. There's a lot of really complicated mechanisms, clearance mechanisms. So whether it gets to the brain or not, you're never going to believe it until you see it in an animal. There's definitely places for pre-animal testing, but unless it gets to the brain of the animal, it can't go into a human for testing. So that's what we did here. We take a mouse and we inject these molecules into the bloodstream, and we fluorescently label them. And then after 7 days, we sacrifice the animal and we ask, where is the molecule we injected? And so here you can see we take out the liver, the lung, the spleen, the kidney, the heart, and the brain. And essentially, if we just take a normal antibody that doesn't target anything, this is the problem of the blood-brain barrier. You get signal in every organ except the brain. Okay, so this is the blood-brain barrier problem. Now, what we did then was we took, for example, here, so first we took the gold standard, which is an antibody that targets a blood-brain barrier protein. This is what everybody does. And after 7 days, we see no signal in the brain. Now we're targeting a novel target. We're just going to call it target X for today. And what's striking is that if we target this, we get very strong brain signal after 7 days, suggesting that this molecule is getting to the brain. It looks like it's getting into the brain, and it's long retained in the brain. Now, this is very attractive for treating something like Alzheimer's disease, where you'd like to deliver molecules and have long retention so you could have long activity of those molecules. Now, if we switch, and I'm just going to show you what we're doing here. So without being too complicated, this Y structure looks like an antibody, and we're just attaching a little extra piece to the antibody that gets it into the brain. And if you do this, we call that a shuttle. So we shuttle the green protein, whatever we're interested in. We shuttle that in by attaching a little extra piece that binds to something at the blood-brain barrier. And so what happens is if we do this with a conventional gold standard, we get no signal in the brain. But excitingly, if you do this now with our novel shuttle, we get very strong retention of this molecule, delivery of this molecule to the brain. So this is really exciting because, again, the sky's the limit in what you could deliver. If you wanted to deliver something that targets beta amyloid or tau, you could put that antibody as the green protein and deliver it. Or if you want to treat something, say inflammation, that reduces inflammation, you could deliver an antibody that does that, and so on. So this is really encouraging, and that's kind of the first step we've taken now in terms of delivering. Now, one of the biggest questions we always get is, how do you know you actually get into the brain? And I can just tell you a long story, seeing is believing. So I showed you at the organ level, but now if you take cross sections of the brain and you look for the blood vessels, so these are blood vessels, these spaghetti things, those are the actual blood vessels of the brain. And what happens is if you deliver an antibody that just targets the blood-brain barrier, this is all you get. But what you really want is this. You want something that actually gets into the brain and actually targets things in the brain. So here we've delivered an antibody that's specific, in this case, for a certain kind of brain cell. And what's amazing is we get very strong transport into the brain and then into brain cells shown in green. and none of it is co-localized with vessels. So we got to the blood vessels, we got across the blood vessels, and then we got into the brain and target things in the brain. So we've done this now with several antibodies showing this approach is general. And so now we're really very excited about this. This really now enables testing of a lot of existing molecules we haven't been able to test because we haven't been able to get them into the brain reliably or at concentrations that are really going to be effective. So that's problem one. So the other problem that I want to talk about is what would you do if you could do this? So if now you can deliver molecules to the brain in an effective way and an efficient way, what would you do? And there's a lot of things you could do, but I'm going to talk about one today. It's not targeting tau or not targeting amyloid. That has a place, but obviously a lot of us think that there's got to be other approaches beyond that. And so what we're interested in is trying to harness the neuroprotective mechanisms that already exist in the brain, that we already know that if we could stimulate some of these certain mechanisms, this could be potentially therapeutic in Alzheimer's disease. So the long and the short of it is, we know of this certain protein. It's a neuroprotective protein. It has a fancy name, BDNF. It's not important for today, but we know that there's this neuroprotective protein in the brain. And we know that what it does is it binds to neurons, and there's a certain receptor on the surface of neurons. And when it does, it causes this cascade effect, and it makes the neurons happier. It makes them sort of alive and signaling these neuroprotective pathways, and that neurons do better when this molecule is around. It's been known for some time. The problem is this is not a drug. So if you're in drug development, just because something has an activity does not mean it can be made into a drug. For example, if you stick it in the bloodstream, sometimes those molecules are degraded, they're cleared very quickly, they have very poor biodistribution, they have all kinds of different potential problems. This is arguably one of the worst drugs ever, BDNF, and people have tried to use it in clinical trials. It's the worst. But what if we could get the same activity as we get from that protein, but now in the form of a molecule that's a drug, that's a validated drug that's already been used. At least the format of the molecule is what I'm trying to say. So that's what we want to do. We want to replace this natural protein with an antibody. So antibodies have now been validated for treating cancer and lots of things. And so what we want to do now is say, could we make an antibody that binds to this receptor in exactly the same way and activates the receptor the same way as the natural protein, but now in a form where you could actually imagine developing that into a drug. So that's what we're doing. So we're taking an antibody in green here that we validate in our lab, activates this receptor just like the natural protein does. And we're attaching our little domain here that gets us into the brain. So now that we can deliver this neuroprotective molecule to the brain and then stimulate this neuroprotective signaling. So this pathway is independent of tau or A beta. So this could be a complementary therapy. We think probably where the world is going is combination therapies, where you're going to have to hit more than one pathway for neuroprotection. So for example, the beautiful work we heard about in the last presentation potentially could be combined with this, because this doesn't affect tau or amyloid pathology. That's the interesting thing. It provides neuroprotection through independent mechanism. And that means that when we think about mouse models, so we have to be careful. Mouse models are a critical part of the drug development process. You have to decide when do they make sense and for what. So we're currently starting some of these experiments where we're using a tau mouse model. The tau models are better than the beta amyloid models in mouse. And we're asking a very specific question about whether we can provide neuroprotection in the setting of these mouse models, where we're targeting conserved pathways that are conserved from mouse to human, the blood-brain barrier transport conserved from mouse to human, this neuroprotective signaling pathway conserved from mouse to human. And we're really thrilled to get started with this, and we just want to thank you all very much for this incredibly generous support and your encouragement and your inspiration, and it means so much to us. And I want to thank the people that do the work, and really, I have to say, in science, one of the things that is so exciting to learn about really is what goes on behind the scenes. And these people are really the ones, so this is a PhD student, his name is Mew, and he was just the absolute inspiration behind this entire project. He got the critical first results where we knew we were on to something. And he's a very hard worker, a fun guy. He's a ping pong, very good at ping pong. He's on the University of Michigan ping pong team, and he went to the international competition in Texas, or the national competition in Texas recently. And then Mike is a postdoc from UT Austin, so from down south, and Lane is at Michigan-Ann, so they're doing a lot of the animal experiments. And then Mike, Jennifer, Nealon, and Wade are involved in making these molecules that we're trying to deliver. Thank you very much for your attention.